Well, thanks everyone for having us. It's great to be here. The weather's great. It's not hot and muggy. All right, so I'm going to try and just explain some high-level concepts that we have about diagnostic imaging and performance data, especially in terms of quantitative imaging. So here's actually I stole this from Nick, but here's the basic diagnostic imaging assessment. We have the patient, then you have your imaging system, physician, often radiologist. I think we're biased because we're the Division of Radiological Health, so we just say radiologist, but there are lots of physicians using that. And then they're using those images to get at some kind of concept of interest or diagnosis for the patient, something they want to look at. So as Robert mentioned, the validation really depends on the claims you're making. So for a medical device, this depends on the intended use or indications for use. I can know a little bit about the biomarker qualification for drugs. There's also a biomarker qualification for medical device development tools. And in that, they have a different term. We call it a context of use. But it means similar things like, what are you describing? What is your tool? What is the biomarker doing? So the validation depends on it. So again, what's the evidence required? Well, it depends on the intended use. So we'll look at it differently. So the way I was thinking about this was, well, in this example, it's kind of easy to see where's the device. Okay, and the imaging system. But in terms of validating the concept of interest, I have all these other elements. So, well, I can't control much about my patient. They come with all their different variability or disease conditions. Well, I can characterize my imaging system. I know what the outputs are. I know what the inputs are. Get some idea what it is. Well, now the physician's going to use this information. So now it's getting in a little trouble. Like, well, how do I validate my physician? Well, different physicians might look at the same data and make different decisions, which might lead to different outcomes. So where do you validate then? So we know that the acquisition and analysis settings impact the results and interpretation. So as I mentioned, we can understand the imaging system, but it has a lot of knobs and whistles and bells and tools. So, and as Robert mentioned, these often have tool-type indications. They're indicated for things like cross-sectional area of the body. But even the same CT scanner, the same MR scanner, you can use it and get slightly different quality images or vastly different quality images. And most of these are Class 2 devices regulated through the 510(k) process. So we ask that sponsors provide validation consistent with their claims. Tool type, as Robert mentioned, they permit device manufacturers to make medical devices available to the community faster. And that allows more innovative technology to be in the hands of the physician, treating patients, diagnosing patients. However, those tool type claims encourage clinical testing for specific intended uses that aren't called out in the indications for use statement by the clinical community. So the CT scanner, when we looked at it, it can produce cross-sectional images of the body. Can you use it in lung cancer screening? Well, you might need some additional data for that. Can you use that for radiation therapy planning for some type of specific tumor? Well, you might want to look at that very carefully. And as I said, many of these imaging devices, these diagnostic, they have many things, tools, different pulse sequences, different CT scanner settings, different reconstruction settings. They all make it a little different. So it's very hard to validate exactly what you want. So how is it? Now, when you move slightly to a different domain where you get to more quantitative imaging, well, it gets a little more clear about what the medical device is and where we expect the validation, because it has a clear numeric output. We can characterize this statistically. We can look at things like reproducibility, bias, linearity, and really get a good performance assessment. There's still that radiologist at the end, so different physicians, radiologists, oncologists, they might look at the same number and interpret it differently, but you get a better understanding of where it is. So when we go to quantitative imaging assessment, kind of consider this whole chain. So we also know, as I've been mentioning, that there's many, many factors that influence reliability. So this is true for the tools as well as for biomarkers, whether you're using it in clinical practice or in clinical studies. One way that we've seen successfully is you can kind of control the variability you have. So even if you don't understand it, you can limit it. So, okay, we're only going to use scanners that have 128 detectors. We're going to use these types of settings. And that encourages a little bit more consistency, controls your variability a little bit. So that's what you can do in the context of a clinical study. In clinical practice, every hospital is different. Some Hospitals, at their individual hospital might have kind of top-down settings. So at the hospital, everything looks the same, but across hospitals, you get, your patients started out in California, moved to Chicago, moved to Florida, went different centers, different systems. Data is going to look a little different. So as I mentioned, there's things you can't control, like the patient, all types of variation in physiology, anatomy, lesions. There's acquisition settings like protocol, slice thickness, exposure reconstruction, the imaging platform, manufacturer. And then there's the measurement. How automated is it? Is it semi-automated? Does the software algorithm matter? Are you using the same software version? The software manufacturer might have made a change in the software version from version 20 to 26. Do you know how much that influences your marker? So in order to understand that better, we look at different types of evidence. There's phantom evidence, both physical and digital reference objects, or digital phantoms. You can use simulations, realistic models, clinical data, reader studies, all these to help validate imaging. So as I mentioned before, for tool-type claims, though, kind of the message I would share from my portion of the talk is who validates the output. So this became so apparent to me recently that a manufacturer put this in their indications for use. So this is a cleared 510K. And they had this statement that says, which I've just been reminding everyone, I'm sure you all knew already, the accuracy of the determined physical parameters depend on the system and scan parameters. So that must be controlled and validated by the clinical user. So also, quantitative imaging, this can support other medical product approvals, whether this is kind of devices or drugs, biologics. So you can use that, and you can validate that then in the context of the clinical study that it's used for. There's all types of uses for imaging, radiological imaging, and clinical study, from inclusion-exclusion criteria, endpoints, patient stratification, safety-related endpoints, dosing, et cetera. So I think it's a really wonderful thing, but encourage validation. So just to highlight two things, there's two different biomarker programs at FDA. There's the Medical Device Development Tools program that's still in pilot, but hopefully to be moving to full program sometime in the near future, whatever near means in terms of government. And there's the CDR Biomarker Qualification Program. Both of these rely on the concept of context of use. And so I just include the definition. I'm not going to read it. You can look it up. It's a resource, and I have a couple other resources. Here's what we consider the definition of biomarker. Both these definitions also come from the BEST glossary. But a biomarker, we're really looking at a kind of a defined characteristic that's measured as some indicator of normal biological processes, pathogenic processes, responses to exposure intervention, including therapeutics. And this includes radiographic characteristics. However, a biomarker is not an assessment of how an individual feels, functions, or survives. And we're not part of CDR, but those three words are very important in terms of drug development. So just how important is imaging in terms of the biomarkers? Well, approximately a third. I'm off a little on my math, I know, but about 6 out of the 19 that have been made public are imaging-based, including SUV of tumors, SPECT, hippocampal volume for MR. So there's a lot of interest in imaging-based biomarker qualification. Dr. Califf, who's the new commissioner, he always highlights this resource, so I think it's a good idea. If you want to know definitions related to biomarkers, there's this BEST glossary. It's called Biomarkers, Endpoints, and Other Tools. This is the outcome of an NIH FDA working group that apparently had a lot of back and forth over months, but it was published back in January. It's on the National Library of Medicine website. It's a living document, can be updated. I think they take comments and stuff, but it has a lot of great definitions. Definitions really help in terms of understanding and having clarity when we're talking to different people. In terms of how to validate kind of imaging biomarkers, a really great resource is the KIBA metrology papers. This is a series of 5 papers. There's another one in radiology. That's the Quantitative Imaging Biomarker Alliance. It's a subset of RSNA. FDA has been involved in, and I think has co-authorship on all those papers, but they talk a lot about different concepts related to how to validate quantitative imaging. So I think that's all I had.